Good morning, students. In previous class, we were discussing about food fermentation and the process of food fermentation. In today's class, we are going to discuss about production of lactic acid. When we talk about the properties and chemical reactions of lactic acid, firstly, the structure of lactic acid. It includes 3 carbon organic acid where at one terminal there will be carboxyl group. On the other terminal, there will be one hydrocarbon group, and in the central carbon atom, there will be an alcohol carbon group. It is soluble in water and water miscible organic solvent. That's why it having low velocity. There are various reactions are present which are characterized with lactic acid and which undergoes with esterification with organic acid and dehydrogenation or oxygenation to form pyruvic acid or its derivatives. This is the structure of lactic acid in the isomeric form. Now, there are different applications of lactic acid. It uses as acidulant in vegetable and leather tanning industries. In small scale application, it also works as pH adjustment, termination agent for phenol formaldehyde resins, etc. Then it having pharmaceutical and cosmetic application also in the tropical ointments, lotions, anti-acne solutions, etc. Calcium lactate is used for calcium deficiency therapy. It is biodegradable polymer, so it is applied in medical science also as sutures, orthopedic implants, etc. Polymers of lactic acids are biodegradable thermoplastics and their degradation can be controlled by adjusting the composition and the molecular weight. It having the similar properties as petroleum derived plastics. They are high boiling, non-toxic and degradable components. Now, the uses of lactic acid. It used in the bakery industry. Originally was produced to replace tartrates in baking powder with calcium lactate. Later it was used to produce calcium stearyl 2 lactylate, a bread additive. In the medicines, when we go for the calcium deficiency therapy, so in that one case, to introduce calcium in the body, it provided in the form of calcium lactate. Esters of lactic acids are also used in the food industry as emulsifiers. Lactic acid is used in the manufacturing of rye bread. It is used in the manufacturing of plastics. Lactic acid is also used as acidulant I have discussed previously. It is non-volatile, odorless and is classified as generally regarded as safe by the FDA. It is a very good preservative and pickling agent. Addition of lactic acid aqueous solution to the packaging of poultry and fish increase their shelf life. Now this is the whole process that how polylactic acid is used in the plastic production. As we have already discussed about the glycolysis process, citric acid cycle, and you know very well that in our body we having aerobic respiration and fermentation also. Then what is the difference between the aerobic respiration and fermentation? So this is the whole process which perform in our body where the aerobic respiration takes place during lactic acid in cytoplasm and how in the molecules or the particular forms are going to include in electron transport chain. When we talk about the production of lactic acid at industrial level, then for that we having homofermentative lactic acid bacteria named as Lactobacillus species especially L. delbrueckii. In recent time, Rhizopus ericea has been used. Both organisms produce the L form of the acid. Rhizopus fermentation much shorter in duration and isolation of the acid is much easier. Lactic acid is very corrosive and the fermenter. They are sterilized by steaming before introduction of the broth as contamination with thermophilic Clostridia yielding butanol and butyric acid is common. Such contamination drastically reduces the value of the product. Now, this is the whole process, that how the chemical synthesis occur and how the microbial synthesis occur. In the chemical manufacturing, the protochemical resources are there that form acetaldehyde, then addition of HCN and catalyze that converts into lactonitrile, then it is hydrolyzed by H2SO4 and the racemic formation of lactic acid will be there. Whereas in biological manufacturing there will be renewable resources will be there, then pre-treatment, due to the pre-treatment there will be fermentable carbohydrate prepared, and after that microbial fermentation takes place in the fermented broth, then recover and purification takes place where the pure L and D lactic acid is present. Lactic acid fermentation are of two types. One is homofermentative and one is heterofermentative. In the homofermentative, as sole product by reduction of pyruvic acid with the help of the enzyme lactic acid dehydrogenase takes place. One molecule of lactic acid is formed from one molecule of pyruvic acid. In heterofermentative, products are lactic acid and ethanol or acetic acid and carbon dioxide. They produce lactic acid from one half of the glucose molecule and ethanol or acetic acid and carbon dioxide from the other half. The rod-shaped lactic acid bacteria are distributed in several genera, though majority are placed in the genus Lactobacillus. These are the names of homofermentative bacteria and these are the names of heterofermentative bacteria. How the conversion of glucose get converted into lactic acid or the fermentation takes place due to the lactic acid bacteria. Then in homolactic there will be conversion of glucose 6-phosphate to fructose 6-phosphate, then 1,6-diphosphate, then it will form glyceraldehyde or hydroxyacetone. Glyceraldehyde will again convert into 2-pyruvate and 2-pyruvate will get converted into 2-lactate. This is the whole process which is included when there is when we have studied about the glycolysis cycle. Then heterolactic. In heterolactic process there will be glucose get converted into phosphogluconate, then it will convert into ribulose 5-phosphate, then cellulose 5-phosphate will be formed, then it will convert into glyceraldehyde 3-phosphate or acetyl phosphate. If the glyceraldehyde 3-phosphate is present over there, it will convert into pyruvate and then convert into lactate. Whereas in acetyl phosphate, it will convert into acetaldehyde and get converted into ethanol. When we talk about the microbial culture, Rhizopus oryzae converts starch directly into amylolytic enzyme activity. Lactobacillus delbrueckii converts hexoses into lactic acid in fermenter at pH of 5 to 6.5 at the temperature of 46 degrees Celsius to 60 degrees Celsius. When we talk about the Lactobacillus bulgarius, it converts hexoses into lactic acid in fermenter at pH of 6 to 7 at the temperature of 43 degrees Celsius. When we talked about mixed culture of Lactobacillus brevis and Lactobacillus pentosus were used to produce lactic acid on large scale production. Then this is the process that how we can extract lactic acid. or recovery is a main problem which is related to the fermented lactic acid production. When we go for the lactic acid production, the production is takes place very easily, but it is very difficult to extract it. So lactic acid is crystallized with great difficulty and in low yield. At the end of fermentation, when sugar content is about 0.1%, the spent medium is pumped into settling tank, where it go for the calcium hydroxide at pH 10 is mixed, and mixture is allowed to settle. After that, clear the calcium lactate is decanted off and combined with filtrate. Then we treat it with sodium sulfide, decolorized by absorption with activated charcoal, acidified to pH 6.2 with lactic acid and filtrate. The calcium lactate liquor may then be sprayed dried. Now, when we talk about the citric acid production, then commercial importance of organic acid. When we talk about the organic acids are used mainly as food acidulants. The main organic acid in industry uses citric acid, acetic acid, tartaric acid, malic acid, lactic acid, etc. Compound of citric acid, iron citrate, is used in pharmaceutical industry. Organic acids are obtained as the end products or as the intermediate component of a particular biochemical cycle. Citric acid is used as a suitable and cheaper replacement of polyphosphates in the detergent industry also. It having the properties of anti-foaming agent, as a softener, and in the treatment of textiles also. Now, when we talk about the citric acid biosynthesis, citric acid was produced by Mucor and Penicillium species as fungal metabolite in media limited and phosphate. Presence of citric acid was detected as a byproduct of calcium oxalate produced by a culture of Penicillium glaucum. A great number of problems had to overcome before an effective fermentation process could be used commercially. At present, over 90% of the world's output of citric acid is produced using the process of microbial fermentation. Nowadays, citric acid is produced by the fermentation technology, which is used by the filamentous fungus Aspergillus niger, mainly through surface and submerged fermentation of starch or sucrose based media. In industries, there are byproducts are also available which are totally waste, and nowadays at industrial level we are using different bacteria to use that one waste to produce citric acid. These are the some examples given under. What are the industrial applications of citric acid? Citric acid has a long list of applications in industrial sector, in foods, in beverages, in pharmaceutical industries. Lots of uses are there. It mainly used as a preservative, antioxidant, chelating agent, and acidulant. It is flavor enhancer, plasticizer, and synergistic, as well as sequestering agent. Its rising demand is subsequently causing an increase in global production. Its pleasant acidic taste and is high soluble in water. That's why it is majorly used in the food industry. Well, in our previous classes when we discussed about the citric acid cycle, in that one you know very well that we have discussed about there was the formation of citric acid from acetyl CoA, and then it was converted into isocitrate. So it is the intermediate process and intermediate molecule. Other than this, it was discovered that Aspergillus niger could accumulate citric acid in a media rich in carbohydrate but deficient in phosphate and trace elements like iron, zinc, manganese. The regulation of glycolysis is most important for citric acid production. The fermentation is carried out aerobically in large fermenters, and a key requirement for high citric acid yield is that a medium be iron deficient. This is because citric acid is overproduced by the fungus as a chelator to scavenger on. In next one slides, I have discussed about the whole process, that how the citric acid forms, and how we can go for the synthesis of citric acid from commercial strains. Then single cell proteins are also present over there, and how it works, what are the advantages of using microorganisms, and what are the limitations of single cell proteins, how the production of single cell proteins takes place, which undergoes with the microbial screening, suitable raw material, process engineering, then apparatus technology is also there, safety demand, and on the basis of that different microorganisms are present over there, which include in the single cell protein production, how they perform, what is the comparison of microorganism, how they perform and how much they are advantageous and disadvantageous. Some factors have also discussed which affects the biomass production and how they affect the production of single cell protein, what are the applications of these single cell proteins, and where we can use these single cell proteins in our industries, and it is in use. So this is all about. Just go through with this video, and if having any kind of doubt, please welcome for discussion. Come and discuss it properly and clear your doubts. Thank you.